Welcome back, BIO178. In this last video for our Genomics and Transcriptomics module, I'm going to walk through a case study which is based on my own research because it's what I'm very familiar with. And what I will talk about here is an example of how you can use RNA sequencing to study adaptation in particular. So I'll walk us through kind of the whole process, the background information, the data that was obtained to help us come to these conclusions. And so I'll note that this type of study is different from the Bobcat study I've referenced, for instance. So in that study, they were taking samples from individuals and just comparing what the gene expression response was. But in that study, they didn't do any type of common garden or any type of experimental manipulation to quote unquote even the playing field. So basically the data they're getting is just showing us differences in gene expression in response to these differences in the anticoagulant poison levels, for instance. We can also, though, use RNAseq to help us determine which genes are differentially expressed, not just because of recent environmental differences, but specifically because of adaptation to environmental differences. So those adaptation are things that have been happening evolutionarily throughout generations. And in order to do that, you have to do that common garden type of experiment we talked about previously. So in this example that I'm going to talk about in this video, all of the data was obtained following this common garden period where I collected individuals from the field and then kept them in aquaria tanks at identical conditions before doing the actual experiments. So we'll do a case study here focusing on our, at this point, very familiar friend, the black turban snail. So a little bit of background. I've talked a lot about these sites already, but we have data from the intertidal at these northern and southern California sites. We know that southern California sites are hotter. So in this graph, we have temperature on the y-axis and days. So this is from August through October in 2014, which is generally when it's the warmest. And we can see that for all those Southern California sites in red, they reach much higher temperatures than the Northern California sites in blue. And in particular, if we look at this dotted line here, this skull and crossbones, 41 degrees is the temperature based on some of my other experiments that we know causes 100% mortality for all individuals, regardless of where they came from. And what's important to note is that some of these Southern California sites are already exceeding those temperatures in the field. So that's important to think about in terms of how these populations are going to respond to climate change in the future. So we know that there are those temperature differences, and we also have phenotypic evidence for local adaptation to heat stress. This is another image I've shown previously in the local adaptation lecture. We have percent survival here on the y-axis, different heat stress temperatures that individuals were exposed to. Again, Southern California individuals in red, Northern California individuals in blue. And we see at 3 of these temperatures that Southern California individuals have significantly higher survival than Northern California individuals. Again, this was performed after a common garden period, so we know any phenotypic differences we're seeing are due to local adaptation. And just to point out here, this arrow, this is indicating at that 41 degrees where I had that hash mark on the last graph, we see 0% survival or 100% mortality for all individuals from all populations. So we have this phenotypic evidence for local adaptation, and now we want to think about what the molecular mechanisms are that might be underlying that. So you can think about molecular ecology or the ecological genomics that are understanding this local adaptation to heat stress. And when we think about the potential genetic mechanisms underlying local adaptation, there are 2 possibilities. There can be differences either in gene expression or in gene sequence, or it could be in both. So let's go back to a brief refresher here of the process of gene expression. We start with our DNA that's transcribed to RNA, translated into amino acids, and then folded into our final protein. So it's possible that gene sequence changes can be causing local adaptation. For that, I did a study that we've talked about previously, doing that ddRAD sequencing to identify thousands of SNPs across the genome in both coding and non-coding regions. So a gene sequence change could lead to local adaptation by something like this example shown here, where you have a nucleotide change that is then causing an amino acid change that then causes the protein to fold differently than it normally would. So that can be an example of how a gene sequence change can cause local adaptation. If we think about proteins, if it's an enzyme, that shape of the enzyme is very important in how effective it is and how functional it can be. So if you're causing a shape change in that protein, it can have some large phenotypic effects. Gene expression changes are another example of a mechanism that could be causing local adaptation. And for that, you can look at that using RNAseq, which is what I'll focus on here in this video. So in that example, you might see something like where transcription levels are increased. So you actually have more copies of that particular RNA, which leads to more copies of the amino acids and more copies of that protein. If this protein is something like a heat shock protein that helps the organism survive high temperatures, having more of that protein available can help that individual survive high temperatures better. And again, when we're looking at all of the RNA that's expressed in an organism at some given point in time, that's considered the transcriptome. So for this study in particular, I focused on 2 Northern and 2 Southern California sites. This is basically just a result of budgetary constraints. So in the past, I've always talked about 3 Northern and Southern California sites. Here, for money purposes, we did 2 populations, 2 geographic regions from each region. And here's a quick walkthrough of the methods. So we collected animals, put them in common garden. One group we exposed to a heat stress designed to mimic a typical low tide period that they would see in the field. Low tide when the water is out and they're exposed to air and to the sun. That's when they see the high temperatures in the field. Another group was non-stressed control group for comparison. We then extracted RNA, did library preparation, and sequenced using that Illumina platform I've talked about previously. And then for the bioinformatics or the data analysis side, here is that de novo transcriptome assembly I talked about previously. This is the name of the particular program that we use to do that reassembly of the short reads into the full-length gene fragments. In terms of annotation, to figure out what those different reassembled genes are, what function they have, we use a program called BLAST2Go, which basically compares your unknown sequence to a known database to identify what gene what function it has. And then we mapped reads using the same program to get an idea of gene expression level at these different genes, and used an R package called DEseq to identify genes that were significantly differentially expressed in the 2 different geographic regions. So this is the same image I've shown before, just to walk through. We have the short reads that are reassembled into our full-length sequences and then annotated to determine the function of that gene. So let's look at some of the results. This is called a heat map. If you haven't seen one before, I'll walk us through it. So each one of the columns here is a different treatment group. So northern control, northern heat stress, southern heat stress. Each row is a different individual. Each row is a different gene. Yes, each row is a different gene. And the colors for each of those cells, so this cell, for instance, is giving us the expression level for this particular gene for the northern control treatment group. Because that cell is colored blue, it means it has relatively low expression. So in this color scheme, a yellow color indicates that gene is highly expressed, a blue color means it's very lowly expressed. And what we can do here in this type of heat map is compare the expression levels across the Northern and Southern California populations. So here in this green box at the bottom, what we see are heat shock proteins. So heat shock proteins, again, are often abbreviated HSP. Those are things that refold damaged proteins. So under heat stress, what happens is proteins become damaged and denatured. Heat shock proteins help to refold those enzymes or proteins to their natural conformation so they can be functional again. And heat shock proteins are often denoted by these numbers after the name, which indicates the molecular weight of that particular heat shock protein. So in green here at the bottom, we have boxed the family of heat shock protein 70s. And if we look at the expression patterns between Northern and Southern California individuals after being exposed to heat stress, we can see that overall the Northern California individuals have higher expression of this family of proteins after heat stress, indicated by this more yellow color in their cells. In contrast, blocked in red here, we have a different family of heat shock proteins, heat shock protein 40s. And here we see the opposite pattern where the Southern California individuals show higher expression of those particular heat shock proteins after being exposed to heat stress compared to the Northern California populations. So a pattern like this, where we already know that Southern California individuals have higher survival based on the phenotype data, and now we're also seeing a group of genes that have higher expression under heat stress. What we wanted to know is, okay, what is the function of this particular group of genes, and can higher expression of that group of genes potentially be contributing to this higher survival for the Southern individuals? And again, just to back up a little bit about what heat shock proteins are, they're these molecular chaperones involved in the stress response. So again, if we walk through this image here, under normal conditions, we have a correctly folded protein, but under conditions of heat stress, that protein can become damaged and denatured, so it's no longer in its proper conformation, it can't work right. A heat shock protein comes along. I don't know why it looks like a raspberry in this picture, but a heat shock protein, what they do is they're these chaperones that help that protein be folded back into its original proper conformation so it can work right again. And heat shock proteins use energy in the form of ATP to do their job and do that function to help refold proteins. So going back to HSP40s in particular, I do indeed think that HSP40 expression that's higher in Southern California individuals could be contributing to their higher thermal tolerance. And that's because if we think about the function of HSP40 is what's called a co-chaperone for HSP70. So it's essentially a helper for a helper. So what HSP40 does in particular, it increases the hydrolysis rate of ATP for HSP70. So essentially it allows HSP70 to do its job faster because it needs ATP to do its job. If it can hydrolyze it faster, it can do its job faster and also help refold more proteins. HSP40 also allows HSP70 to bind to more substrate proteins. So HSP40 essentially expands the range of proteins that HSP70 can help to refold back to their natural conformation. So we think this higher expression of HSP40 is helping to increase the thermal tolerance of Southern California individuals by increasing the rate and efficiency and range of proteins that HSP70 can help refold under these high temperature conditions. So the conclusion here, based on all of this RNA-seq data, is that gene expression differences are indeed likely contributing to local adaptation to heat stress, and that's in addition to the outliers that I've also identified from the ddRAD RAD-seq study.